So what we want to do in the course today is look at the variation data that we have in Ensemble, thinking about obviously the different types of variation data that there are and the different types of data that are associated with variance. So what we want to do in this module is think about how we can classify genetic variants, thinking about the different species that have variation data available and the sources of that variation data where Ensemble draws the variation data from. There's a number of public databases. And then we're going to think about exploring the variation data through the Ensemble genome browser. So we're going to be looking at getting variation data through the Gene tab, through the Location tab, which we've both seen both of those tabs before, and then also through the Variant tab, a new place that we are going to explore today. And then finally, we're going to be looking at the Variant Effect Predictor, which is a tool that we have in Ensemble that allows you to begin analyzing your own variation data sets. So the first part of this module will be focusing on using the Ensemble browser to retrieve information that's already stored in Ensemble about known variants, whereas the second part will be focusing upon giving you an option and a tool that will allow you to analyze your own data sets. So we have really two themes to this module. So firstly, we're thinking about variation data and the different types that we have. So firstly, we can think of variance in terms of the size of or the number of base pairs that are affected at a single time. So we can firstly think about small scale variants. So these might be things like SNPs or single nucleotide polymorphisms, also known as SNVs, single nucleotide variants. And these small scale variants are going to be things that affect a small number of base pairs at a time. So maybe one base pair or a few at a time. So they also might be things like small insertions or deletions. So here we can see, for example, if we're comparing these sequences of these two individuals, we can see that there's a deletion in the blue individual underneath here, or an insertion for the blue individual here, or also what we would call a SNP or an SNV, where there's no change in the number of bases at a single position, but we do have a change in the actual base that's there. Then we can think about large-scale structural variants. So these are going to be things like also deletions or insertions, but affecting perhaps hundreds or thousands of base pairs at a time. But it can also include things like duplications, translocations, and copy number variants as well. So in Ensemble, there are 24 different species with variation data available. Obviously, the amount of variation data that's available for a species depends on the sequencing efforts that have been carried out around the world for that particular species. So human being the most well-studied, most sequenced species on the planet, we have nearly 700 million individual variants for human, which we can explore in the browser later on. But there are also lots of, obviously, variants that have been identified across model organisms such as mouse and rat. And then also the agricultural species have been very well studied as well. So looking at things like cow and also horse, pig, and sheep as well, for example. There's also a number of species in Ensemble genomes that have variation data available. So there are many plants, such as Arabidopsis and wheat and barley, for example, that all have variation data available. And then there's also a large number of fungi, and then a few metazoa and protozoa that also have variation data available as well. So what I want to do now is explain to you about the Ensemble variation data pipeline, so how we import variation data from a number of sources, process it, and then add some of our own analysis as well. So we have these four main steps. So firstly, we import data from a range of sources. We apply some quality control when we're importing the data. Then we look to other databases to look for linked or associated data, and then we begin analyzing those data sets ourselves and then making that available to you through the browser and the other interfaces. So firstly, the import of variation data. So we look to a number of different publicly available archives, variation databases where we draw the variation data from. So when we're thinking about human, the most probably famous and most populous variation database is dbSNP. So this is a database hosted by the NCBI, that has many millions of variants stored for human. So we import the variation data from dbSNP in that case. For non-human species, we look to a database called EVA, the European Variation Archive. They have lots of variation data stored for non-human species. DGV Archive is quite an interesting database. It has lots of data about structural variants. And then also finally, we do look to databases such as COSMIC and the Cancer Gene Census to get information about somatic variants as well. So we're thinking about the difference between germline and somatic variants. Databases such as dbSNP and EVA have information about germline variants, but we also do store information and data about somatic variants from databases such as COSMIC. Then we apply some quality control. So we do things like mapping to the appropriate reference genome assembly. So from human GRCH37 to 38, for example, we check the alleles match between the report and what's in the reference genome sequence. And we check the RUPAC ambiguity codes as well. So these are just some standard checks that we perform across all the millions of variants that we import, just to check that they match up with what we expect and what we already have in our databases. The third step is to look for what we call linked or associated data. So for every single variant that we import in step one, we look to a number of different databases to see if there's additional information, such as allele frequency information, so how frequently the different alleles of a variant are observed in different populations around the world, phenotype and disease associations, and even things like publication data as well. So these are the three main associated value-added data sources, data types that we look for, and this is just a snapshot of some of the sources. So for example, the allele frequencies, we look to projects like the 1000 Genomes Project, Nomad as well, and other sequencing projects to get the allele frequencies. Phenotype and disease associations, we look to sources like the NCBI ClinVar databases, and also places like OMIM. And then the publication data, we look to Europe PMC and PubMed. So as I said, one of the main types of linked data that we look for is allele frequencies. So we look to some of these major projects here that I've listed, such as the 1000 Genomes Project, the Nomad Project, UK10K, TopMed, and also the NCBI Allele Frequency Aggregator Alpha Project. And we retrieve the allele frequencies for all of the variants that we have imported in step one. So if any of the variants in our database from step one match up with variants that are identified in these projects, we also then import the allele frequencies and store those in our databases. The phenotype and disease data comes from a range of different sources, and this can be dependent upon the species of interest. So obviously for human, one of the main sources of phenotype and disease association is ClinVar and OMIM. But then we also look to sources like IMPC for mouse, the rat genome database for rat, for example, ZFin, animal QTL for farmed animals as well, for example. And also it's important to note as well the GWAS catalog. So we also import information about variants, that have been associated with phenotypes through GWAS studies and are stored in the GWAS catalog. So we get things like the clinical significance, the inheritance type, reported genes and variants and risk alleles. And then for the GWAS studies, we also get things like the p-value, the odds ratio, and beta coefficients as well for the disease associations. And then the final step, step four, is where we apply our own analysis. So Ensemble predicts firstly the variant consequences. So we look to see the effect of the variants upon the genes in where they might be falling in the genome. We also look at protein function prediction, so whether a variant will be deleterious for a particular protein if it's affecting the coding sequence of a protein, for example, the coding sequence of a transcript, for example. We also calculate linkage to equilibrium, and we look at conservation across species as well by performing some alignments of closely related species. So one of the first things that I said that we do in Ensemble is that we predict the consequences of the variants that we import into our database. So just to quickly summarize the process or how we predict variant functional consequences, is that we take each variant one by one and we plot where it falls on the genome, which is represented by this black line here. And where a variant falls in the context of the genes that are found there will determine its functional consequence. So we can think of a variant, for example, that falls outside of a gene. It might fall in a 5' upstream or a 3' downstream region, in which case that variant might not be directly affecting the protein, the encoded protein structure and function, but it might fall in a regulatory region, for example, which will obviously affect the mRNA transcription level, transcription activity of that gene and its transcripts. Then we can think about a variant that might fall within the gene, but not within the coding region. So it might fall in a UTR region, or in a splice site, or even in the intron. So these variants, again, they don't directly affect the protein structure and function, but they might affect the mRNA stability, the splicing, translational efficiency, and the other factors that might lead to a change in the expression of the protein level. Finally, we can think about a variant that falls within the coding sequence of a gene that's depicted in yellow here, for example. And these come in two main flavors. So firstly, we can think about a variant in the genome that doesn't result in a change in the protein sequence. And these are called synonymous coding variants. And we can get these because the genetic code is degenerate. So we know that more than one codon can encode the same amino acid. So what that means here is that there might be a change in the genome sequence that doesn't result in a change in the encoded protein, the encoded amino acid at that position. So we call that a synonymous coding variant. On the other hand, we might have a change in the genome that does result in a change in the coding sequence, that does result in a change in the encoded protein at that position, which is what we call a non-synonymous or a missense variant. So that's what we do. So for every variant, we plot where it falls along the genome, and then we assign a consequence based on the position where it falls in the genes that are located at that position in the genome. So the consequences that we assign to the variants have very specific terms. So I don't expect you to be able to read this table. This is taken from the documentation you can see at the bottom here. But this is just to explain to you about the different consequence terms that we do assign. So the consequences are all defined by what we call sequence ontology, SO. And just like we had gene ontology, which was a standardized vocabulary of terms to describe gene function, here we have sequence ontology, which is a standardized vocabulary of terms to define sequence changes. So here, as an example, you can see the yellow color coordinated in yellow here. We have the definition for missense variant. So a missense variant is a sequence variant that changes one or more bases, resulting in a different amino acid sequence, but where the length is preserved. So this is the definition of a non-synonymous missense variant. And that's what we can see here. So any variant that has this specific effect, we label that as a missense variant, and you will be able to see in the Ensemble browser that it will be yellow. So the color coordination matches to what you can see on the side here. Obviously, I don't expect you to remember what all of these colors mean, but as you use Ensemble more and more, you'll get to know the colors maybe a bit more off by heart, and there's always a legend that will explain to you the colors as well. So when we're thinking about variants, specifically missense variants, we can think of them in terms of their pathogenicity. So there are various different algorithms that score or rank the changes in the amino acid sequence based on how well conserved the protein is, that particular amino acid is, the chemical change at that specific position where the amino acid is being changed from one to another, and the 3D structure and domains. So basically what happens is, obviously we have a single position in the protein that maybe encodes an arginine, and then because of a variant at a specific position in the genome, that arginine changes to a glycine. So what these different algorithms do is they predict how deleterious that amino acid change will be on the protein structure and function. But it's important to remember that these are predictions. So they're sort of algorithms that compute all of these factors to make a prediction about how deleterious this amino acid substitution will be on the protein structure and function. And then obviously to follow up from that, it's always good to validate these predictions experimentally. So there's a number of different algorithms that try to predict pathogenicity of amino acid substitutions. And I've just tried to summarize them here. So we have all of these scores in Ensemble, and we'll see this in the browser demo in a moment. So we have SIFT, which is a score of between 0 and 1, where 0 is predicted, well, a predicted score of 0 indicates that that change will be deleterious, whereas 1 is more tolerated. For polyphen, the opposite is true. So a predicted polyphen score of 0 suggests that the amino acid substitution will be benign, where 1 is more damaging. CAD works on a scale of 0 to 100, with 0 being benign and 100 being likely deleterious. And then we also have Revel, Meta-LR, and Mutation Assessor as well, which all work on a scale of 0 to 1, with 0 being tolerated or benign, and 1 being more likely to cause disease or damaging to the protein structure and function. So before we start the demo, one of the most important things that I wanted to go through with you now is the terminology that we use for the alleles of a variant. So just to go back to my presentation from yesterday, we can think about a single chromosome, which is made up of contigs laid end to end, just like this. So we discussed yesterday how a single contig actually is the sequence from a specific individual. So you can see here, for example, this chromosome is made up of 4 contigs. Obviously, this is just an example, but each of these contigs comes from a different individual. So if we take a look in more detail at one of these contigs, we can see that this contig has a sequence, and we can identify this position that's highlighted in red here as a T. Now, the lady who was sequenced as part of the Human Genome Project where this contig was derived from, she's not actually very representative of the human population around the world. So the T that lady has is only found in 5% of individuals, but the other 95% of people around the world have a G at that position. And G is also the allele in all primates. So if we were to align this sequence to orangutan and chimpanzee and gorilla, for example, we would find that all of those primates also have a G at this position. And the T that lady has does have some underlying disease susceptibility associations as well. So she might be more likely to develop high blood pressure later on in life, for example. However, she was the woman who had her genome sequenced to derive this sequence of the genome, and so T is the allele that we found in the Human Genome Project. So T is the reference allele, and G is the alternate allele. So it doesn't matter about allele frequencies, it doesn't matter about ancestral conservation, and it doesn't matter about disease susceptibility. T is the reference allele, and G is the alternate allele. And so we would always list those alleles in Ensemble as T/G. So the reference is what's found in the reference genome sequence, the GRCH38 reference genome sequence, and then the G is what has been found in other sequencing experiments after that. So in the 1000 Genomes Project or the NOMAD project, one of these projects, for example. Now, obviously, what we then look to after this stage is to see whether these alleles whether these alleles have frequencies associated with them, and in that case we can assign a minor and a major allele. So in this case, the reference allele is a minor allele, and the alternate allele is the major allele. In many cases, it's actually the opposite that's true. So in many cases, it is the reference allele that is the major allele, but it's not always true. So in this case, the reference is the minor allele. And in this case as well, the alternate allele is also the ancestral allele. Okay, so hopefully that makes sense. I think it's something that I come across quite frequently when I'm teaching workshops, is that there's this common misconception about reference and alternate and minor and major alleles. So it's just very important to keep these definitions in the front of our mind when we're thinking about the genome sequences and variant alleles. Okay, so in our demo, what I want to do is have a look at a single gene called the MCM6 gene. And then we're going to look for the variants that are falling within the MCM6 gene. Then we're going to have a look at the genomic region, so the location tab where the MCM6 gene is found. And then we're going to have a look at a specific variant that has this ID here that we're going to search for. And we're going to find out more information about that variant. So if you're following along in the course book, the demo starts on page 28. That's where the screenshots begin. But you can also follow along with me. So the first thing to do is to open up a tab in your browser and to navigate to the Ensemble homepage, just like we did yesterday. And I'm going to search now for my gene of interest. So the gene is MCM6. And then I can click Go. And then navigate to the Gene tab, just like we did yesterday. So I won't spend too much time going through what we can see on the Gene tab, but just to mention that we have this menu over on the left-hand side where we have these links for all of the different information. And what we're going to focus on now is this section over here on the left-hand side. So firstly, you can see a section here called Genetic Variation. So here we can see Variant Table, Variant Image, and Structural Variants as well. So firstly, I'm going to click on Variant Table. So what we can see here, if you begin to scroll down, is that we have a very, very large table. You can even see my cursor on the side here. It's, you can see this table is many thousands and thousands of rows long. So what we essentially have here is a table that contains information about all of the variants that fall within the MCM6 gene, the short variants that is. And so what you can do is a number of things. So firstly, you can export this table if you want to. So there's an Excel icon in the top right-hand corner, which allows you to export the table as it is, and then you can begin searching and filtering for it outside of Ensemble. Or you can use the filters above the table, just here, just above the heading of the table. So you can filter the table on a number of different factors: global minor allele frequency, SIFT score if you're looking at missense variants, or the same for polyphen. You can also filter to find variants that have a specific consequence prediction, or you can filter on any of the other columns as well. So I'm going to filter using consequences. So if you click on consequences, you'll find a pop-up window that shows you all of the consequences, which are currently all on. So what I'm going to do is turn them all off to start with, and then I'm just going to click back onto missense variant. So now I'm going to filter my table to only show variants that have a missense consequence prediction. And then I can click apply. So now you can see that my table's been filtered. And if you just scan across, you can see in consequence type column, all of the variants are now a missense variant. And you can see exactly as we described before. This is the yellow color coordination that you can see here. And we have the definition when I hover my mouse over the consequence type. So just to quickly show you what we have in the table here. If we pick on this variant at the top, for example, you can see firstly we have a variant ID. So this is a unique ID that refers to this variant itself. And you can see that the number begins with RS. So RS is the variant notation system that dbSNP uses. So you can know that any variant that starts with RS will have been imported from dbSNP. Then we have the location. So this is the genomic location where this variant is falling. And then we have the alleles, T/C. So in this case, we can see that T is the reference, and C is the alternate. So what we're saying is that T is the allele that we find at this position in the genome, in the reference genome sequence, but the C has also been found in some other sequencing projects in some individuals. Then we have the global minor allele frequency. This is actually only calculated for variants that were identified in the 1000 Genomes project. So it's not going to be present for every single variant, only those variants that were identified in the 1000 Genomes project, like here, for example. Then we've got the class, so the type of variant. Here we're looking at SNPs. The source, as I said, dbSNP. And then we have a column called evidence, which allows us to see information about. Then we allow a column called evidence that allows us to see information about different the sources of the variant and the information about the variant. So you can see here, this top variant, for example, has got frequency information, and it was identified in the Nomad project. This variant here, for example, was identified in the EXACT project, the TopMed project, and Nomad. So this is just giving you information about the sources and the evidence associated for each variant. Clinical significance, if that's available, will be shown in this column here. The consequence type, again, we've filtered for missense variant. Amino acid substitution, so E to G in this case. So a glutamate to a glycine at position 820 in the protein. And then we have the pathogenicity predictors, so SIFT, polyphen, and CAD in this case are predicting, well, SIFT predicts that this score will be deleterious. And if you hover your mouse, you can see the color and the description with the pop-up as well. Whereas polyphen and CAD predict that this amino acid change might be more benign or tolerated. So it's important to notice here exactly how these methods work. While they're predictions as well, you can even see that the predictions don't always necessarily agree with one another. So I always think of these as giving us a flavor of what these methods think might happen, but they don't always agree with one another, and they should always be experimentally validated, as I said. There's a question that someone just comes in. I think there's some. Okay, so there's a question here. I think it's from Nathan and also from Nikara as well about the bolding. So I think this is what you're referring to here. So you've got two rows with these triallelic variants. So what I will do is just quickly explain to you now. So here we can see actually that we have two rows that have the same ID. And so these two rows both refer to the same variant. So this is, and it's a triallelic variant. So it's got C/G/T and C/G/T. So in this row we have G, which is in bold, and that means that this row is referring to the consequence of the G alternate allele. So when we change the C to a G, what happens? So it's E to a Q. In the second row, we're looking at the consequence of the T alternate allele. So when we change the C to a T, you can see that actually it's now E to K in the amino acid substitution. And they obviously have different pathogenicity predictions. So that's what the bold allele. So this is just telling you what this row refers to, which alternate allele this row refers to. Okay, very good. What I'd like to do now is move on to having a look at the location tab. So we can go back up to the tabs in the top in the blue bar and click on location. And again, we saw this this interface yesterday, but what we'll do now is we'll add a couple of variation data tracks just so that we can see the variation data and explore it in a bit more detail. So what I'm going to do is click onto the blue configure this page button over on the left-hand side. And then I'm going to click on variation data from the link on the left. So the menu, the subcategories, I'm going to click on variation to show me the variation data tracks. So we can see, as I said yesterday, there's lots of variation data tracks available. So you can show tracks with variants from a specific database, if that's what you want to see. Or you can show variants from the 1000 Genomes project, if you're interested in showing variants from specific continental populations, for example, the African, American, East Asian, European, or South Asian populations. You can also show tracks with variants that have phenotype and disease associations. And you can also show variants from LSDB associated variants, phenotype associations, arrays, copy number variant probes, and also a whole set of tracks looking at structural variants that you can view as well. So there's a number of different databases and consortium with structural variation data that you can turn on as well. So what I'm going to do is I'm just going to turn on a couple of tracks from the 1000 Genomes project. You can see that the Nomad data is already turned on. So we'll look at the 1000 Genomes project to compare. So I'll choose South Asian as an example. So SAS, this is the acronym that we use for the South Asian continental population in the 1000 Genomes project. So we'll click on the track here and turn it on. And then there's a second track underneath with the common short variants. So the variants that were found in the South Asian population at a frequency of at least 1%. So we can see that in the definition with a frequency of at least 1%. So we can again turn that track on as well. So what we're going to see is that the variants in this second track are a subset of the variants in this first track that I turned on. So obviously this is all variants, and this is only those variants that were found at a frequency of at least 1% in the South Asian populations. So once I'm happy, I can save and close. And if I scroll down, you can begin to see the variation data. So we've got the context track with the genes, as we saw yesterday, then some of the other tracks that we also added yesterday as well, including the UniProt alignments. Then we have the Nomads track that's being shown by default. And then we have the 1000 genome South Asian variants, and then the 1000 genome South Asian common variants. So what you can see is that the variants themselves are depicted as these individual lines, which represent the base position where they fall. And they're color coordinated as well. So you can see, for example, these dark blue variants. These are intronic variants. So if we actually look, we can see that these variants that are labeled as intronic are falling at this intron position in the MCM6 gene and its transcripts. So if you want to find out more information about a variant, you can, if you've got a steady hand, just hover your mouse until it turns into this pointed finger icon and then click. And then you can see that in the pop-up window, you get information about the variant. So it's alleles, ambiguity code, allele frequency, and then some evidence sources as well. The final track that I was already turned on by default are the phenotype associated variants. So again, you can see here, these are the phenotype associated variants that fall within the MCM6 gene. Good. Okay. So the final thing that I want to show you in this demo is how to search for a specific variant. So obviously what you could do is you could click on one of these variants, as I just showed you, and click on the RS ID in the pop-up window. Or you could go back to the Gene tab and click on the variant IDs from the variation table. Or if you know the variant ID of interest, you can search for it from the Ensemble homepage or from the search bar in the top right-hand corner. So I'm going to search for my variant of interest just by typing the ID into the search bar in the top right-hand corner. So the ID is RS4988235. And I'm going to copy that for you now and put that into the text box. I can see it's quite busy, the chat box, but hopefully you can find the ID. And then I'm going to press enter. And then I'm going to click on the ID from the search results to navigate to the Variant tab. So again, you can see now we're in the Variant tab. We have a new menu on the left-hand side. And so what we're going to be exploring is information all about this specific variant. So what we can do is see firstly that we're in the Explore this variant page. This is a summary page showing us the most severe consequence, the alleles, so G/A/C. This is a triallelic variant. Then we have information about its position, its VCF notation format as well. You can see the evidence icons that correspond to this variant. You can also see this variant in HGVS format, which is another variant notation format for describing variants. You can see its synonyms in other databases. You can see the genotyping chips where this variant can be mapped to. And you can even see this variant's original source as well. So you can go out to dbSNP if you click on this link. I can do that now. And you can see this entry in the dbSNP database as well. So you can cross-reference the information in Ensemble and dbSNP. Good. So what I want to do now is explore some of the options in the menu on the left-hand side, just to show you some of the information you can get about the variants. So firstly, I'm going to click on Genes and Regulation. What I'm actually going to do is I'm going to hide all of these options that I expanded just now, because otherwise we won't be able to see our data on the page very clearly. So what we can see on this page is a table that shows us all of the predicted consequences of this variant. So what we're going to see here is that a single variant can actually have more than one predicted consequence. And that's because obviously a variant can affect more than one transcript of more than one gene at a time. So what we can see actually is that in this table we've got 6 rows, and each row refers to a consequence that's affecting the MCM6 gene. So in fact, this variant's only affecting one gene in this case, the MCM6 gene. But the MCM6 gene has 3 different transcripts, and we are looking at a triallelic variant. So what we're actually saying here is that we're predicting the consequence, which we can see in this column here, when we are looking at the A alternate allele for this transcript of MCM6, then the C allele for this same transcript of MCM6, then we're looking at the A allele again for the second transcript and the C allele for the second transcript, and then the A allele for the third transcript and the C allele for the third transcript. So these are obviously the predicted consequences. So this variant is falling in an intron of this transcript, and it's also being defined as a non-coding transcript variant as well, because these transcripts here, these are non-coding transcripts. Further down, we also have information from the GTEx portal. So we have information about the gene expression correlations in different tissues as well, which you can explore in your own time if you want to. The next thing that I want to show you is the population genetics. So you can click on population genetics on the left-hand side. So what we're showing you on this page is information from a number of different projects that have information about the allele frequency of the alleles within this variant. So at the very top of the page, we have the 1000 genomes project data. But as you scroll down, you can also find the Nomad, the NCBI Alpha, TopMed, UK 10K, and the Gambian Genome Variation Project as well. So these are the projects that we have allele frequency data for this variant. So let's just have a look at the 1000 genomes as an example. So the pie charts show us a good representation. It's a good representation of the allele frequency. So we've got firstly a pie chart showing us the frequency of the alleles across all populations. So the G is found in 84% of alleles, and the A represents 16%. That's across all populations. And in the African, we have G representing 97 with A just 3%. Then we've got the American, East Asian, European, and South Asian populations as well. If you want to see a continental population in more detail, so for example, if you're interested in the European populations, you can click on subpopulations underneath the pie chart, and you'll open up a new set showing you those European populations. So here we have Utah residents with a Northern and Western European ancestry, Finnish, British, Iberian, and Tuscany populations. And you can see the allele frequencies observed in each of those populations as well. I always quite like showing this example because you can actually see the difference in the allele frequencies across different populations. So in East Asia, for example, in the East Asian populations, 100% of the observed alleles were G, whereas in the European populations, it was basically a 50-50 split between the G and the A alleles that were observed. So I think this is quite a nice example. And what we're actually going to see later on is that this variant is associated with lactase persistence, lactose intolerance. And so what we can actually do is plot the spread of dairy farming around the world based on the prevalence of alleles that are associated with lactose intolerance and the allele frequencies. Underneath the pie charts is a table that shows us something very similar to what we saw in the pie charts. So we have a table with all the populations from the 1000 Genomes Project on the left-hand side. Then the alleles, the frequencies represented as a bar chart. Then we have the allele count in the first two columns. So obviously humans are diploid organisms. So every person who was sequenced in the 1000 Genomes Project contributed 2 counts to the allele frequency. So we have 84%, which is 4,200 counts for G, and A observed at 16%, which was 808 counts. Obviously, as I said, the people who were sequenced in the 1000 Genomes Project were diploid, obviously. So what we also have is information about the observed genotypes of those individuals. So we can see that there are 75% of people that were homozygous for G, around 8% of people homozygous for A, and then just 16.5% of people that were heterozygous AG. The final thing to show you is if you click on the show link on the right-hand side in the genotypes column, it will jump you to the bottom of the page. And you can actually find a table that shows you the observed genotype. So in this case, homozygous for A, in the different individuals in the project. Obviously, these individuals are not named. These are the IDs of the individuals themselves from the project. But what you could then do from this stage is with this, if you're interested in studying perhaps a cell line that is homozygous at this position, you could go to the 1000 Genomes Project pages and order a cell line that's been derived from this specific individual. Or if you're interested in people who are homozygous for G, you could obviously choose the corresponding ID and order a cell line from the data banks in the 1000 Genomes Project. Good. Okay. So the final thing that I want to very, very quickly show you is under Phenotype Data on the left-hand side. So here we can see all of the phenotypes that are associated with our variant of interest. So here we can see the different phenotypes on the table on the left-hand column, the sources of the association. So we can see that the GWAS catalog has made a lot of these associations here between the variant and these specific phenotypes, but also lactase persistence at the bottom, so lactose intolerance, which is an association that comes from ClinVar and OMIM. So from this table, you can see the evidence of this association, so the PubMed IDs that have made this association where this is stored in ClinVar, and then you can also link out to the link in OMIM, and you can see the allele that's associated with this specific phenotype. So the A allele for this gene is associated with the lactase persistence. Finally, if you click on the phenotype itself in the left-hand column, it will take you to a new tab that we haven't seen before called the Phenotype Tab. So from this place, you can see that we have information about all of the features the variants and the genes, for example, that are associated with this specific phenotype. So in this case, we have 1, 2, 3, 4, 5 individual variants that are associated with lactase persistence. And they actually all fall in the MCM6 gene. Obviously, what I've done is I've gone on a very specific route through the browser. So I started in the Gene tab, then the Location tab, then I searched for a variant, and then I looked for the phenotype. But obviously, what you could imagine is if you started off from a place where you were interested in a particular phenotype, you could search for this phenotype from the home page. So you could type lactase persistence into the search bar on the home page to come to the Phenotype tab, and then you could begin clicking on these RS numbers to go out to the Variant tab and begin exploring in the reverse direction. So the way that you use Ensemble will obviously depend upon your starting point and your point of interest, and then you can search from there and explore the different tabs as we've done today. The exercises are all on page 9 and 10 of the question book. So from the training materials, you can click on the question book link. And in the question book, you can move down to pages 9 and 10 and find the exercises here. So we have V1 and V2, which are looking at variants in human. And then V3 is looking at a variant in mouse. Then we also have exercises on rice, fungi, and then also there's an exercise looking at the variation structure viewer as well, which I didn't actually show you, but you can explore as well in your own time. So the Variant Effect Predictor that we affectionately call the Vep is a tool that allows you to analyze your own variants. So the idea is that you come to the Vep with a list of variants that you've already generated yourself. So you might have generated this list of variants from a number of different methodologies. So most commonly, you might have performed some sequencing yourself and then performed an alignment and variant calling process to identify the variants within your sample against a reference genome sequence here that you can see in black here, to identify a list of variants that you will have stored in a file. Or you might have performed a genotyping array, or even you might have searched through the literature and identified a list of variants that are of interest to you. So Ensemble doesn't provide the tools or the platform or the infrastructure to perform these sorts of investigations. So there's no alignment and variant calling software available in Ensemble. The idea is that you do this upstream and then you come to Ensemble with your list of variants that you want to begin analyzing and finding out more information about. So with the Vep, the idea is that it's a tool that you can use to predict and annotate the functional consequences of your variants. So the idea is that you give the Vep your variation data in one of a number of different formats. So they might be a set of variant coordinates, or they might be VCF file format. So this is variant calling format, a very common file format that's used for variant callers. You might have HGVS format, which is very commonly used in clinical variant reporting. You might have a list of variant IDs, such as a list of RS numbers, like we were discussing in the previous demo. And SPEDI as well is another variant notation format that you can use with the Vep. So what the Vep will do is it will take your list of variants, so it will take them one by one, and for each of them, it will map that variant on the genome in the context of the genes, the Ensemble genes that we have annotated in that region, and it will make functional consequence predictions against the gene, transcript, and protein sequences. You can also use the Vep to retrieve pathogenicity predictions, such as SIFT and polyphen scores. One of the other things that you can do is you can ask the Vep to see if any of the variants that you have input in the first step match to any of the variation data in the Ensemble databases. So if the variants are known, or if they're novel. So if the variants are known, it means that the variant that you've inputted matches up with one of the variants in the Ensemble database, and therefore you can retrieve things like frequency data and literature citations as well. Obviously, if your variant is novel, there won't be these sorts of phenotype associations and frequency data available, but the Vep will still process those novel variants to give you the functional consequence predictions and pathogenicity scores as well, for example. You can also get regulatory consequence predictions and splicing consequence predictions as well. So this is just to remind you about the process that the Vep will do once you submit your data. It will take each of your variants one by one, just as the Ensemble variation data pipeline does as well. It takes the variants one by one, plots them on the genome, and then predicts the functional consequences using these standardized sequence ontology terms to describe the functional consequence of the variants where they fall in the gene. So if you want to use the Vep, you've got three main options. So the first one is to use the web interface. So this is a really nice point and click interface that allows you to upload a small amount of data to begin processing and analyzing in the website itself. Obviously, you don't need to have any coding experience to use the web interface. Then we also have a standalone Perl script. This is a tool that you can install and run from the command line. So if you're familiar with the command line and happy to sort of explore the Perl script, it does provide you with a lot more options and more flexibility as well. And it's much better for large data sets as well. So if you perform some whole genome sequencing, for example, and you have a list of 1 million variants, then the standalone Perl script will be able to much more efficiently and flexibly annotate your variation data set. And then also we have the REST API. So if you are familiar with coding in programming language such as Python or R, you can use the REST API endpoints as well to query the Vep. So the method you use will depend upon two things. Firstly, the size of your data set, and secondly, your familiarity with the command line and different programming languages. So what we're going to do today is just use the web interface to show you the principles of the Vep and to show you how it works. And then I can point you towards the documentation for these other methods if you're interested in using those as well. So what we want to do in our demo is to have a look at just a set of 4 variants that have been identified on human chromosome 9. So the deletion of an A at this position, and then 3 different SNPs. So a C to A, a C to G, and a G to A at these different positions. And we'll use the Vep to see if the variants have already been identified. So whether they're already annotated in Ensemble, so whether they're known or novel. We'll find out the functional consequences of the variants and which genes that those consequences refer to, and whether the variants affect any gene regulatory features as well. So in the course book, the screenshots start on page 35, but again, you can follow along with me. So before we start our demo, what we're actually going to have to do, if you do want to follow along with me, is to just get our small data set to copy and paste into the Vep at the right point. So in the page with the course materials, hopefully you still have this page available. You can click on the link here that says copy sheet. I'll put that link in the chat box for you. And you can see here, this is just a copy sheet with some data sets for the different exercises in our course. So here we have a section called Vep Demo. So all you need to do at this point is to copy these 4 rows of data, and we will be pasting those into the Vep at the right point in the demo. You can see obviously that we have some other data sets for some of the other exercises tomorrow. Okay, so now going back to Ensemble, I'm going to navigate back to the home page using the logo in the top left-hand corner. So you can see in the blue bar, we have a link to the Vep tool. So if you click on Vep, it will take you to a page where we have lots of information about how to use the Vep. So we've got the web interface, which we're going to launch in a moment using the button here. But you also have the documentation for the command line tool and for the REST API as well. So you can explore those in your own time if you want to. But we're just going to launch the web interface, so we can click there. And this will take you to the Variant Effect Predictor web submission form. So There's a number of different options that we're going to go through together, and then we can run our Vep queries as one. So the first thing to do is to select your species. So Homo sapiens is selected by default. That's exactly what we need, but you can change that by clicking on the box here if you have variation data from a different species. It's important to note here that the assembly that we have selected, or that is selected for us, is the GRCH38 reference genome assembly. We can't change this at this point here. So if you do have data relating to the human GRCH37 assembly, you'd need to go to the GRCH37 archive and open the Vep from there. The next thing to do is to give our job a name. So I'll call this example, but you can obviously choose whichever name you want to give your job. And then the next thing to do is to upload your data. So before I paste the data into the text box, you can see that underneath the text box, there are buttons that will populate the text box with example data in different formats. So if you're not sure about the formatting of the different data types, you can click on these options and it will automatically fill in the box with some example data. This is not our data, but just to try and demonstrate what the different formats look like. So that can help you in formatting your own data sets. So here, in the text box, I'm going to now paste my data. And what you'll be able to see is that this data is in the Ensemble default format. While you paste that in, I'll just quickly explain the format to you. So you can see that we have 4 rows of data. So each row refers to one of our 4 variants. And we have a white space or tab separated file format here. So the first column is the chromosome number, which is 9. Then column 2 is the start position, and column 3 is the end position. Now, obviously, we're looking at 4 variants that each affect a single base pair. So the start and end coordinate are actually the same in this case. You can see here. It's just the number is duplicated in columns 2 and 3. Then in the next column, we have the alleles. So we have the reference / alternate, so A/- in this case, where we're substituting the reference A for a minus allele, as a deletion. So we use the minus as the allele here. Then we have the C to A, the C to G, and the G to A alleles. The next column is not required, but this is the strand. So this is the strand that these alleles refer to. So in this case, we're talking about alleles on the forward strand. So if you don't put a plus here, the Vep will automatically assume that you are talking about the forward strand. But you can use a minus if you are specifically talking about the reverse strand. But we're talking about the forward strand, so we use a plus. And then the final column is just a name that I've given to each of our 4 variants, var 1, 2, 3, and 4. We're using the text box because we just have a small variation data set, but you can also upload a file if you have a VCF file, perhaps with a set of variants, or you can provide a URL. So if you have a really large VCF and you want to attach the file remotely, you can save it in a public directory and then provide a URL. So the question from Anna that I can answer at this point is how many variants the Vep interface can load. The answer is there's no limit. If you want to upload a file, the limit is 50 megabytes. But if you provide a URL using a remote file, there's actually no limit on the size of the file. The thing that's the limiting factor, I would say, is the fact that when you're viewing the results in the web interface, it can become, the table is very, very large and it can become quite difficult to work with. So you might want to export the table directly from there. So it really depends. So, I mean, you can, my advice would probably be to have a go with your variation data set and see, but there's actually no upper limit apart from the usability of the web interface with a large data set. The next thing to do is to choose your transcript database that you want to use. So either the Ensemble GenCode transcripts, or alternatively, if you want to analyze your variants in the context of the RefSeq transcripts, you can select that transcript set as well if you want to. But we'll use the Ensemble GenCode transcripts as our example here. After that, there's a whole set of additional configurations that we can select, and they're hidden underneath these expandable menus. So I'm firstly going to click on identifiers. And here we can see that we can already, well, the Vep has already selected some of these options by default for us. So for each of the variants, we're going to find the gene symbol, so the name of the affected gene that the consequence refers to, and also the transcript version, so the stable ID of the affected transcript. We can also get any CCDS IDs, protein IDs if we want to get the Ensemble ENSP IDs of any affected proteins, and also the UniProt IDs as well if you want to get the IDs from UniProt, from affected proteins. You can also choose to get the variation data reported to you in HGVS. So in this way, the Vep can be quite a nice tool for reformatting your data. So if you have a list of variants in VCF format and you want to convert it to HGVS format, you can select this here, input your variation data in VCF format, and retrieve an output of HGVS formats. Equally, as we saw above here, you can input variants in HGVS format, and then out of the other end, you'll be able to export the data in VCF format and get the RSIDs as well. So the Vep can actually even be thought of like a variant recoder, where you can input one format of variation data and get another format out the other end. The next section is called variance and frequency data. So the first option is asking us if we want to find co-located known variants. By default, the answer is yes here. So what the Vep will do by default is it will look in the Ensemble database and see if there are any co-located known variants. So information about the variants that we've uploaded. In which case, if there is a matching variant, then we'll be able to get information from the 1000 Genomes project or the Nomad project, for example, as well, and also get the PubMed IDs and citations. Now, just to point out to you as I go through these options, I'm just picking a few of them to show you how they work in principle, but you can obviously go through this in your own time and think about exactly which options you want to select in your query. The next section is called additional annotations. So here, again, by default, we're going to find the biotype of the affected transcripts. We're going to find the transcript support level, so the TSL score of the affected transcripts. And we'll also see the APRI annotation status and whether the affected transcripts are the main select transcripts as well. We can choose the amount of upstream or downstream sequence that will be, the distance upstream or downstream of a gene that the variants will be reported against. So in this case, if our variant falls within 5,000 base pairs upstream or downstream of a gene, it will be reported as affecting that gene of interest. We can also find the IDs of any affected protein domains within the encoded proteins that are affected, and we can get regulatory region consequences and phenotype data as well, if we want to. In predictions, we can see that there's pathogenicity predictions. So by default, we're going to get the SIFT and polyphen score, but you can also see that we have DB, NSFP, CAD, Condel. These are other pathogenicity predictions as well. Splicing and conservation, you can also see if there are any splicing or conservation predictions for your variants if you select these options as well. And then finally, we have filtering and advanced. So in the filtering, what you're able to do is you're able to pre-filter your data before the analysis to exclude variants that might be common, for example, if they have allele frequencies, or you can perform some advanced filtering to search for genes of interest or consequences of interest. My advice, though, is probably not to do any filtering at this stage, to analyze your full variation dataset, perform the annotation on the full dataset, and then you can begin filtering and prioritizing your variants after that, so using the output of the Vep. But you can filter if you want to, but I'll still stick with no at this point. The advanced options just allow us to increase the speed of the Vep query if we have a really large data set, but these standard settings are okay for us at the moment. So what I'm going to do now is click the green Run button. Oh. Okay, so maybe, hopefully, this is just an intermittent problem. If you got the same issue, then you can press Run again, and hopefully it will work for you. So this will take you to a page with with all of your Vep jobs. So you can see that this page is refreshing every 10 seconds. And here I have my job, Vep analysis with the job name here, example, and it's currently queued. So hopefully it will be now submitted, and then when the page refreshes, it will be running or done. So from this page, you can save your jobs, you can edit and resubmit your query, you can share your results, or you can delete them as well. So if you want to save your data, if you want to save your job, you'll need to create an Ensemble account, which you can do with the login/register link in the top right-hand corner, just as I'm highlighting at the moment. The Ensemble accounts are free to create, and it really just creates a workspace for you where you can save your analyses, you can bookmark pages and those sorts of things. So if you don't log in and save your job, then it will be deleted in a few weeks' time. So if you're going to be running lots of Vep jobs and you want to save the analysis for looking at again and again later on into the future, then I would advise you to log in and save your jobs as well. Okay, so once hopefully you're all at the same stage as me with your job listed as done. So then you can click on View Results. And that will take you to the web into the results page. So at the very top, we can see some summary statistics. So we have a table that just explains some of the Vep job. It's a summary of the Vep analysis of all the variants combined. So we can see that there are 4 processed variants, which corresponds to our input, which is okay. We haven't filtered any variants out. And of those 4 variants, 1 was novel and 3 are existing. So it means that 1 of them has not been found in the Ensemble database. It doesn't correspond to any known variants. So that's a novel variant, but 3 are already known to the Ensemble database, and we will have retrieved all of the allele frequency, phenotype associations, and literature citations associated with those. Our 4 variants have affected 2 different genes, which actually is 7 different transcripts and 1 regulatory feature as well. Then we have some pie charts that show us the consequences of the variants combined and the coding consequences as well. As we scroll down, we can see the results table, which I want to talk through in just a moment. But before we do that, you can just see these options in the table heading. So firstly, you can navigate. If you have many results, then you can scroll across to see all of the results in a number of, it's paginated in a number of pages. You can also filter the table at this point now if you want to. So you can begin filtering on any of the different columns. So you could say filters, consequence is, and then choose your filtering term. So maybe missense variant, and then add. So now you can see that my results have been filtering only to show me missense variants. So this is what I was trying to explain before. It's best to do your analysis on the full data set, and then you can begin filtering and prioritizing afterwards. I want to remove this filter, so I'm just going to click on the Remove Filter option just so we can talk through the full results set, but that's just to demonstrate how to use the filters. Then finally, over on the right-hand side, you're given options for downloading the data. So as you can begin to appreciate, this table is really quite large with lots of rows and lots of data. So it might be easier, rather than viewing the table in the web interface, you can export the data in VCF, Vep or text format, and then begin manipulating the results outside of the Vep in your, on your machine using a number of different tools. Okay, so when we look at the table, what you can see is that we have our uploaded variant is in column 1, and the variant number isn't ordered in the way that we put it in. So what I'm going to do is click on the grey arrows in this column so that we first are looking at variant 1 and then variant 2, 3, and 4 going down. So my variation data is now ordered in the order that I input it. And now we can begin exploring across. I won't talk about every single column, but just to highlight some of the main features to you. So firstly, we have the genomic location. So this is where the variant is found. You can actually click on this to go to the location tab and see this in the genomic context. The allele, this is the minus. So this is the deletion, the deletion of the A that we had as variant 1. And we can see that this is an intronic variant of the COC4 gene. So this is the transcript that this consequence refers to. So this variant is an intronic variant of this transcript of the COC4 gene, which has got this stable ID. However, it's also a downstream variant of this transcript of the COC4 gene. So what we can actually probably work out here is that we have two transcripts of the same gene, one being much longer than the other, and then the variant falling at the position in the genome where it's falling in the intron of one of the transcripts, but that's actually downstream of perhaps the much shorter transcript. So, and it's the same for this transcript as well. So this is actually, this variant is also an upstream variant of this transcript of COC4. So COC4 has all of these different transcripts, and the variant will have varying consequence predictions based on where it falls in each of those transcripts. As we begin to scroll across, we can begin to perhaps see some of the data. Obviously, you can see we have lots of empty fields here because this particular variant wasn't falling in any coding region, so there's no exon numbering to report, there's no HGVSP to report, there's no cDNA, CDS, protein position, or amino acid to report either. As we scroll down, we can perhaps get to see a bit more of this data in more detail. So here we have variant 2. So this is also an upstream variant of the COC4 gene, of this transcript of the COC4 gene, and of this transcript of the COC4 gene. But it's also a synonymous variant of this transcript of the true B2 gene. So what we're actually seeing here is that the COC4 and the true B2 genes are probably located very close to one another in the genome, and the variants falling right where these two genes are either overlapping slightly or perhaps closely located to one another. So it's having an effect on the transcripts of the true B2 gene, but also effects on the transcripts of the COC4 gene as well. And we can also see that variant 2 is falling in a regulatory region as well. So it's falling in this promoter that has this stable ID. So we're going to think more about the promoters and regulatory features tomorrow. But you can click on these links, the stable IDs, to go to the gene, the transcript, or the regulation tabs. Variant 3, we're going to explore in a bit more detail. So you can see that variant 3 is actually a missense variant of these transcripts of COC4. So this variant's falling in a coding region of these transcripts of COC4. So when we begin to scroll across, we can see that it's in an exon. It's in exon 2 of 7 of this transcript and exon 2 of 4 of this transcript. These are the HGVSC and HGVSP notation formats. We've got the position of this variant and the amino acid change, so a serine to a cysteine that's encoded by this triplet codon here, TCC to TGC. Then we have a column called existing variant. So this is quite interesting here. So in this column, we have the RSID that corresponds to the variant that's already stored in the Ensemble database. So where this column is blank, this means that there is no accession number that's already associated with our variant of interest. So this is actually our novel variant here. And then the variants with an ID in this column are going to be the known variants. Then we also have information about the feature strand, the main status, the transcript support level, APRIS annotation, as well as ENSP. Now, hopefully, we also have the protein IDs from UniProt, so you can click on these protein IDs if you want to. And you can also see the SIFT and polyphen scores as well. So SIFT and polyphen are only applicable for missense variants. So we only have these predictions corresponding to the missense consequence predictions. And then finally, we have the domains, the affected protein domains, the allele frequencies from the different projects, Nomad and 1000 genomes. And then we also have information about the associated phenotypes for the overlapping features. So hopefully what you can begin to appreciate is that the Vep will firstly give you the consequences of your variants, but it will also retrieve a wealth of other data about your variants of interest, which you can begin to use to process, filter, and analyze your variation data sets. Good. So that brings me to the end of the demo for the Vep. So what I want to do is give you an opportunity to have a go at the exercises. So what we have, it's now 3:45 in the UK. So what I think we'll do is we'll have 15 minutes to have a go at the exercises for the Vep. You can also, if you get through those, you can go back and have a look at the variation data exercises from the previous section today. So that will take us to around 4 o'clock. In 15 minutes time. We'll come back together to see how you've all got on with the exercises and see if there are any final questions. And then we'll have a 10-minute break again, just to give you another break before we go on to the comparative genomics section at 4:10. So what I want you to do now is have a go at the exercises. Again, you can find those in the question book. So there's just 2 Vep exercises here on page 11. So there's Vep V7 and V8, the exercises. So V7, you'll need to make sure that you put the variation data in the correct format. So this is not the correct Vep notation format, so you need to make sure that you reformat the data when you're submitting your Vep job, and then select the right options to get the answers to the questions here. And then V8 is an example using a VCF input notation format. So you can use this as well as an input to answer these questions.